Hi, welcome to my talk as part of the Biological Sciences Lily Batten Postgraduate Researcher event. I'm Callie, one of many postgraduate research students within the school, and I'll be talking through my research journey and what my research here involves. My research journey started at school, where I knew I liked science, largely because of my interest in animals, and biology was always my favourite science. I wasn't exactly sure what my favourite part of biology was or what I should continue with afterwards, so I decided at university to just study biology as a whole. For my undergraduate degree, I studied biology, and I spent the first few years figuring out what it was exactly that I really enjoyed the most in biology. The labs we did, such as the experiments shown here, were all very enjoyable, as was learning interesting things about plants, and all of this led me to choosing all of the optional plant units in my course. Not only this, but it led me to pursue a placement in plant sciences. I was lucky enough to do a placement year at the Royal Botanic Gardens Kew, and as you can imagine, it was a beautiful place to work as I was able to explore the gardens year-round. Here I was able to do my own research project. I spent most of this time looking down a microscope, but not just a regular microscope, also these really high-powered electron microscopes. For example, here you can see a transmission electron microscope. It looks a bit like it belongs in a spaceship. It took some pictures a bit like this one on the right here, and also a scanning electron microscope which took pictures like this one on the left. On the left is a picture of some stinging hairs of a nettle, and on the right is stomata, which is essentially a gas pore of a leaf. It was really inspiring to work with my supervisor, who was an experienced female researcher in plant sciences. She helped me learn how to become an independent researcher, and after the year, we published a paper on my work. I then came back for my final year of university, where I did a final year project looking at the pollen of Arabidopsis, which is a model plant that we use a lot in plant research, and then graduated almost 2 years ago. This now brings me to the present day. Now I'm here at Bristol as a postgraduate student doing a research master's. But what exactly does that involve? As you probably guessed from the running theme of this presentation, my research now is in plant science. But why plants? Well, what do you think plants do for us? I'm sure you could give me some ideas. Well, here's some ideas of what other people came up with. There's some really obvious ones here, like food, oxygen, medicine, that you might have come up with. But there are many more things that aren't on this list that you may have thought of. This is just to show that there are many reasons why plants are great. But what if we can make them even better at what they do? We've actually been trying this for years already. For hundreds of years we've used traditional breeding to change crops over time. By crossing the best crops, we've actually created crops hugely different from their original ancestors. For example, the banana used to be a much smaller, dull green fruit that was packed full of seeds, quite different from the bright yellow seedless banana we know today. Corn is another example of a crop that's changed hugely. It used to consist of only a handful of tiny, hard kernels on a very short stem. Today we have these massive corn on the cobs that consist of hundreds of bright yellow kernels. Though traditional breeding has worked for us in the past, there are some issues today. It's quite a slow process crossing these plants, and unfortunately is a very random process. Mixing two plants and mixing their genes is a bit like mixing a deck of cards. You shuffle them all together and you get what you pull out. Rarely do you get the aces or the exact hand that you want, and it's the same with breeding. Unfortunately, traditional breeding is not going to keep up with our rapidly growing population. So we need something new. This is where plant biotechnology comes to play. This is the modern day of plant breeding, a process that directly alters plant DNA without the need for crossing plants and hoping that the right crosses work. These changes could be adding new DNA, removing DNA, or making changes in the present DNA. These are tiny changes in a plant that can induce a really big change. We do this a lot in research to find out the effects of particular genes on DNA. But this also can be applied to the real world. We do this via two main methods. The first, using a bacteria. This bacteria naturally infects and alters plants' DNA to insert its own. So instead, we provide the bacteria with the DNA that we want it to add. Unfortunately though, this bacteria can only infect a limited number of plants. The second method uses a gene gun, which fires DNA carried on tiny particles at very high speeds to break through the plant cell wall and into the plant cell, where it then deposits the DNA. As you can imagine, this can be quite damaging to the plant, and therefore is quite inefficient as many of the cells die. This is where my research comes in. My research looks at developing a new way to do this, using a nanotechnology-based method created here at the University of Bristol. I work with a group of women at the University of Bristol, both staff and other postgraduate research students, who are all contributing to our knowledge on this method. This method uses carbon nanodots, a nanoparticle, to carry and deliver DNA into plants. These were chosen because of their ability to pass into plant cells naturally without the need for force, and we've modified them to carry DNA into the cell where it is deposited. They look a bit like this diagram. Blue is the carbon dot, green for the little modifications that allow DNA to bind, and the yellow represents the DNA rings. This method is actually very simple as mixing these nanodots with the DNA and spraying them onto plants. Here's a couple of images from my research so far. Quite complicated to look at, but without going into too much technical detail, I'm looking to help improve the efficiency of this technology. I'm currently using gels and looking at cells under microscopes. The gel, pictured on the left, can help us see how far DNA travels with and without the carbon nanodots, potentially telling us how well they bind together under different conditions. The microscope helps us visualize green fluorescent protein, a green protein that we are using as a marker to show us which cells have been given the DNA. So now that you know a bit more about this technology, you might want to know why we're doing this. Let's go back to the start and think about the many things that plants do for us in the world. There are endless applications of this technology, and we can use these tiny changes in DNA to help solve some really big problems in the world. For example, we could look at the issue of climate change. There's a lot of changes that climate change are making to the world, such as drought and heat. We can make plants that are able to adapt to these changes more readily, for example making drought-tolerant plants. But we could also make plants that help us combat climate change. For example, we can make plants with enhanced photosynthesis, and this makes them more efficient. With more efficient plants, we don't need as much farmland, which means we don't need to have as much deforestation, and then that reduces the impact that deforestation has on the world. Secondly, we could look at food security, a really important issue with a growing population. We can make crops that produce more or better crops, or even change the nutrition within these crops. We could even remove allergens like gluten, potentially creating gluten-free wheat. There is even the possibility of making plants that produce important molecules like medicines or vaccines, which uses plants as little biofactories. We could even use them to make edible vaccines. As you can see, there is really great potential for plant biotechnology, and this is why I'm really keen to research more into this area. I hope you enjoyed this little talk about plant biotechnology and my research journey. Hopefully you now appreciate a little bit more about how all these small changes can be used to solve some really big problems in the world. Otherwise, I hope you enjoy the rest of this event today, and thanks so much for listening.